Hi everyone, welcome to the Bioinformatics Coach channel. In this video, I'm going to show you how to add gene names to variants in a VCF file. So let's get started. Now, before you reproduce this tutorial, it's important to make sure your PC satisfies these requirements. You need to have a Linux or macOS system, and you also need to have these tools installed: bcftools, bgzip, and tabix. All right. Linux or macOS, these are Unix-like operating systems, and they are actually very used. But let me just put it: they are heavily used, and they are actually the preferred operating systems to use when analyzing bioinformatics data. All right, they have utilities that allow you to process files in an efficient manner. You should also have BCF tools. We'll use BCF tools to add the gene names to the variants in the VCF file. And by the way, the input files we are going to use, I have them prepared, so I'll show you where to download them. So no need to use your own data. After that, you can practice with your own data. We will also use bgzip. We are going to use bgzip to compress one of the input files, and then we are going to use tabix to index that input file that we compress using bgzip. All right, so don't worry, just make sure to follow, and I'm going to talk about these tools later on. So let's get started. Now, for this tutorial, I was able to get the instructions for this tutorial by visiting this page. I will shift to that page now. So I got the information I needed by going through the post in this page. All right, so specifically this particular part here. Okay, so I'll leave the link to this page in the description box in case you want to get that information. So I'm actually very grateful that this solution was given. So I'm basically going to reproduce this here. Okay, now let's proceed. Now we need to get our input files. I'm assuming that you already have your system set up. I repeat, I'm assuming that you already have your PC set up, so you are ready to go. So we have this input files. We will need two input files. We need a bed file which has the coordinates as well as the gene names. All right, so coordinates, I'm referring to chromosomes, and then the region. Okay, each gene will have a coordinate. So the bed file here has that information. So we are basically going to tell BCF tools to get the information from the bed files and then use that in the VCF file, which is this one. So these are the two input files. Now with the bed file, let's just look at it before we even download. So I have it something like this. Okay, I will explain more about the bed file, but first let's download these files and we will talk about them, and then I will proceed. Okay, so the link to this page is in the description box. I repeat, the link to this GitHub repo here is in the description box. So just check the description box, get the link, and we can start. I will start with the first one here, the gens.bed file. All right, so to download, click it, and when you click it, you'll be sent to this page here. All right, so when you are here, there are a couple of ways to download. I am going to download via the terminal, and so I'll need a download link. So for the download link, I'll come to raw, this particular button here, and then I'll right click, and then I'll copy the link. Okay, I'll right click. I'm just showing it again. I'll right click and then copy the link. Then I'll move to the terminal, and then I'll start. So on the terminal, this is a new project, and so therefore it's important that we create a folder or directory for it so that we don't end up modifying other files on our system. So I'll first say make dir, and I'll say variants, or you can use any other name, that's fine. I'm going to use variants, and then I'll cd into it. All right, and then I'm going to download the input files. We are starting with a bed file. Okay, we have already copied this download link. All right, so I'll use wget, and then I'll paste the link. There's the link. All right, and then I'll run it. So when I run it, it's a small file, so it's going to download quickly. I have it downloaded, so I will just clear the screen. I will do an ls, and I'm going to find the bed file here. We will talk about the bed file later. Now let's get the other input file. So we need the VCF file, which is also here. Let's get the VCF file. All right, so there's it. It's already gzipped. So I'll just click it, the same procedure, click, right click, copy the link, all right, and then move to the terminal. And then I'll use wget to download it. You can also download using your browser, by the way. Let's download with the link. So I'll paste and then I'll download it. All right, it's also done. Let's check it out. I'll do ls and I'm going to find the file also here. I repeat, you can download using your browser. So at this point in time with the download, I will leave it up to you. So choose whichever method is appropriate for you. All right, but if you're working on a server, a remote server without any graphical interface, then definitely you have to use the terminal to do it. All right, okay, good. Now let's start. First of all, we will need to do a bit of exploration first. I like us to do this so that beginners can appreciate and they understand what we are doing. All right, now this is the bed file which has information, okay, that we will need. Okay, so this information will now be added to the information we have in the VCF file. Now, let me say that if you perform variant calling, all right, a lot of the tools by default will not include the gene names, but sometimes you may want the gene names to be added for your own reasons, maybe for statistical reasons, maybe for other reasons. Okay, and so that is why you have to use tools like bcftools that allow you to add the gene names to the VCF file. All right, sometimes you may be interested in variants in a particular gene or genes. Okay, so the quickest way is to do the annotation, add the gene name so that you can extract that information from it. So there are a couple of reasons, but for this tutorial, the focus is just to add the gene names basically. All right, so let's proceed. So to add the gene names, you need two inputs, the bed file and then the VCF. So the bed file has information which bcftools will use, okay, and then populate the appropriate field in the VCF file. So let's look at what a bed file is. Bed files are fundamentally text files, okay. So you can ignore the extension .bed. They are text files, so that means you can do a head and then check what you have in there. Okay, so this is a bed file. Now, it's important for you to know what a bed file is. So let's go to this Wikipedia page and look at what a bed file is. So this is the bed file format. I will link the link to this page also in the description box. So it's a text file that is used to store dynamic regions as coordinates and then associated annotations. So annotations, basically, these are information. All right. Now with the bed file, let's scroll down. We will have the formats. So with the bed file, there's the format. We have the first column, second column, and then third column. These columns, the first three columns, these are mandatory. All right. And let me say bed files are tab separated. All right. So for bed files, these three columns, the first three columns are mandatory. The others, they are optional. All right. Let's just confirm from here. You can see the information here. Yeah, good. Okay. So yeah, these ones are mandatory because every information you add, okay, it needs to correlate, okay, it needs to be aligned to the first three columns. So we have the chromosome, okay, so for example, chromosome 3, chromosome 4, chromosome Y, etc. All right, so you need to have that chromosome, okay. So whether chromosome number or chromosome name, well, it depends on you, but you need to follow the best practices. So here I'm using the chromosome numbers. Okay, for the chromosome, sometimes you can have it as CHR3, or you can just have the number itself. So it depends. All right. Now in my case, I'm using just the numbers. So take notes. Okay. So we have chromosome 1, for example. Okay. So this is for the first column. Now the second column, we have start coordinate on the chromosome. So that means that in the genome, where does the chromosome start from? Okay. And let me just rephrase it. So that information you are adding, okay, the chromosome that you have, which parts of the chromosome, okay, and then which parts again, that's the end. So start and then the end. All right. So the chromosome that you added here, which region of that chromosome are you representing, okay, in that bed file? That is what we mean here. Which region? It can be everything, or it can be some parts. All right, so the start coordinate on the chromosome or scaffold. Okay, so here it's also important to know that some information, some bed files are 0 based, some are 1 based. So you need to make sure to check that information as well. Okay, so it's important. Okay, so here it says the first base on the chromosome is number 0, i.e. the number is 0 base. So that means this is 0 base. But always you need to just confirm, check with whoever gave you that information, that bed file, to make sure that you are dealing with a 1 base bed file or 0 base. Okay, in this tutorial, we are going to skip that information. This is just an example, but if you are going to use a bed file, just make sure to ask that information. Okay, because that will have an impact on the information in the gene. All right, so the chromosome starts and the chromosome end, the end coordinate. So this chromosome here, which region? So that means you have to give the start and then the end. If let's say the chromosome has a length of let's say 1 MB, okay, let's say the length is 1 MB. All right, now which part are you representing? Or let's even use absolute number. Let's say the chromosome, the length is let's say 1000. Okay, which part are you representing? So if I'm representing, let's say, from region 100 to 200, then the chromosome start is 100, chromosome end is 200. So these three are mandatory. It's important to always include them. Then you add the other information: name, score, strand, fixed, start, etc. So in this tutorial, our focus is to add the gene names to the variants in the VCF. So the bed file contains the column number 4 as well. All the other columns, they are not parts. Okay, so we have chromosome start, chromosome end, and the name, that's the gene name. All right, so that is what you saw when we opened the bed file. Let's just get back here. So genes.bed. So that's what you saw here. So we have the first column, chromosome, start, end, and then the gene name. So a gene will be found in the chromosome, but then the gene is part of the chromosome. It doesn't occupy, or let's say it doesn't form the entire chromosome. Okay, so since it doesn't form the entire chromosome, you need to get its coordinates where in the chromosome. That is what we have here and then here. So start and then end. Okay, so it's always important to take note of that. So that's what you have here. All right, so that's the bed file. And let me also say that with the bed file, this one, I extracted the information from a GTF file. Okay, so if you have your reference sequence, you can get the GTF or the GFF format and extract that information into a bed file and then use that. All right, so it's something you can use some Linux commands to do that, maybe awk or sed or whichever. Okay, there are commands that can allow you to do that extraction. So that is for another tutorial. Okay, so this the bed file in a nutshell. All right, so that's what I just did. So I just the head, we have it here: chromosome, chrome start, chrome end, and then the name. Okay, the gene name, we have that here. Now you realize that for the chromosome names here, I have 01 up to I think 14 or so. I didn't add a CHR. Okay, I just used the numbers here. I'll just display just the chromosome. So that's the first column. So I'll just say cat d $C F1. This is a command, okay, in line of that allows you to extract certain fields in a text file. Okay, and I'll just say genes.bed. Okay, I'll execute this command. Now this command is just for demonstration, so you don't need to run this particular command, but if you still want to do it, that's fine. Okay, so I'll run it and I have lots of chromosomes here. Okay, I have a lot of them. Okay, now let's clear the screen. I'll just want to get just the list, and also includes duplicates. So I just want the chromosome names themselves. So I'll do this. I'll just say this and I'll add sort, and I'll say unique. So this will add the chromosomes, okay, for us. So that's what we have. And by the way, so this particular data is variants that I called from a Plasmodium falciparum isolate. Okay, so that's what I'm using. Now the Plasmodium falciparum isolates, aside these 14 chromosomes, there are other ones here. So we have API MIT. Okay, so that's why you have these ones also here, because I just extracted everything from the GFF file. Okay, so just take note of that. Okay, so let's proceed. So basically that's what we have in the chromosomes. Okay, so let's do an ls and then let's proceed. Now, if you go to the VCF file that you have, the chromosome names, okay, it should be the same as what you have in the bed file. Okay, if you want the annotation to work, you should have the same name. So here we are going to use this opportunity to also look at how to view the chromosome names, okay, in a VCF file. And we are still using bcftools to do this. Okay, so just make sure you have bcftools installed. Okay, so I'll just do it this way. So let me just say, to get the chromosomes, the list of chromosomes in a VCF file with bcftools, you say bcftools query-f. I bring my single quote, I'll say percentage symbol here, that's a shift5 chrom/n. And then I'll specify the inputs VCF file, which is this one. So let's run this. Yeah, so we have them here. Okay, now I'll clear the screen. I'll just rerun, but this time I'm going to remove duplicates. So I'll add my pipe and I'll say sort and I'll say unique. So I have them here as well. Okay, so this is the chromosome list. So you realize that this here, the naming convention is the same as what we did in the bed file. Okay, we have them here, the numbers here. Okay, so that's what you need to note here. Now here, even though in the bed file I have this API and MIT sequences, it doesn't really matter much because we can't find them in the VCF file anyway. So at this point in time, it doesn't matter. What is important is that the information in the VCF file, they should be the same, okay, in the bed file. All right, if there are extra information in the bed file, it doesn't matter. bcftools will ignore that. So just make sure that information in the VCF file and the bed file, they are all the same. That's what you need to know. So what is here, whatever is here in the VCF file should be in the bed file. Okay, that is if you want to get accurate information. Or let's say put information that you want to be added to the VCF file in the bed file. All right, so that's what you need to know. So if, let's say, you are interested in annotating just maybe chromosome 1 and 2, then in that case, you can generate a bed file that has information for just chromosome 1 and 2, and bcftools will use that here. So all that's important here is that whatever information that you would like to be added to the VCF file, you make sure you put it in the bed file. That is what you need to note here. Okay, now let's proceed. Now we've talked a bit, I've done some intro, so let's move on to action. Okay, now let's do this. Let's clear the screen. Let's get our files again. Okay, now we are about to start the show. Now, with the requirements for annotating, or let's say requirements for adding gene names to a VCF file using bcftools. Okay, if your input file is a bed file, then you need to make sure this bed file is zipped. So you have to make sure it's in the bgzip format. That is why I had to add bgzip as a requirement, a two requirements. So we need to bgzip first. And so what we need to do is to say bgzip and then specify the file that we are going to bgzip, which is gens.bed. So I'll say gens.bed, and I'm going to run it. Now, when I run it, if I do an ls again, you realize that the bed file has now been gzipped. That is what we have here. It's now zipped. So we now have it as gens.bed.gz. All right, that's what you have here. Now, for gzip files, if it's a text file that was zipped, you can still use zcat to just read it. This is just a demo. Okay. Just to see some information. You can use zcat to view the contents, okay, of a zipped text file. All right, so that's just by the way. Okay, now let's proceed. So we have our bed file gzipped, or let's say bgzipped. Now I'll need to index it, so I'll use tabix. So I'll say tabix, and I'll say gens.bed. I'll run it. Now, when I run this tabix tool on the bed file, the zipped bed file, if I do an ls, I'm going to find another file here, the index file. Okay, so we need to make sure we have done these steps before we now start the gnames addition. All right, so these are requirements, these are prerequisites. You need to make sure you do that. Okay, now it's time to add the gnames to the VCF file here. Now, before we do that, it's important to check the contents of the VCF file, okay, to make sure that everything is okay. Now, with what we are going to do, we are going to add the gnames and it will go into the info field. All right. Now, for those who don't know the VCF format, just check online. I'll leave some links you can use to check the format of a VCF file. All right. Now, we will need to check the info field because that is where the gnames are going to be added. So what we are going to be doing now is this. I'll say bcftools. I'll say query-f, single quote, and this I'll say percentage info. The info should be uppercase /n, and I'll say snips.vcf.gz. All right. So let's run this. Now, I just want just a few of the lines, so I'll just do a pipe and say head. All right. But in principle, you can do this if you want to display everything, but I'll add this just so we get few lines. Okay, I'll do it this way. Now, let's run this. When you run it, this is what you are going to get. So this is what is currently in the info field. All right. So after the annotation, we expect that we will have another entry for the info field. Okay, so there will be an entry which has the gen names. All right, so that's what you need to note here. So we will get back to the gen names addition. But it's important to first examine your VCF file to make sure that they are okay. Okay, sometimes you can have the VCF file and it would have already been annotated. So in that case, you don't need to add the gen names. In our case, we don't have it, and that's why we are adding the gen names. Okay, now let's clear the screen. Let's ls, and then we can proceed. Okay, now it's time to add a gen name. So to do that, we will use bcftools. Now the bcftools, if you are going to use it, we need to use the annotate command. All right, so let's see this. We are going to use the bcftools annotate command. So let's check it here. So there's the bcftools manual page. This is the annotate. So edit VCF files, add or remove annotations. So that's what we are going to use. You can click it to look at the options available. All right. So that's what you need to note here. So here we are going to use a couple of the options. We will use -a, we will use -c. We're also going to use -h. That's the header lines. Okay, so these are the ones here. So the explanation for all the options I'm going to use are already here. So we have -a, -c, and then -h. So just visit this page and read more about it. The link to this page is in the description box. So take note of that. Okay, so -c, let me just start from here. So -a, that's the annotation file. All right, so the file that contains information which has to be, or which should be added to the VCF file. So you see here it says bgzip compress and tabix index file. That's what we've done. All right. We also have the columns here. So the columns here you have, it says comma separated list of columns or tags to carry over. Okay, so basically you have information that has to be carried over basically. All right, so we have that also. So let's get to the -h. That's header lines file. Okay, so lines to append to the VCF header. Okay, so it's important. So this will be appended to the VCF header. All right, so headers, these are lines that begin with hash. They are not variant records, but they are information that can be added. Okay, so we have everything we need. So let's proceed. Let's get back to the terminal and then let's proceed. So we have this. So to add gnames to a VCF file using bcftools, we need our inputs file, which is bgzip and has also been indexed using tabix, and we have the index file here. And then we need the VCF file itself. All right. So we will start. So we will say bcftools annotate, say -a, that is the annotations file. So here it's gens.bed.gz, and then -c, so the list of columns to carry over. So the -c is supposed to be uppercase. All right, uppercase. So we have chromosome, that's chrome, from, to, and then a gen. So if you take a look at our bed file, let me just open it here. Let's go to GitHub. You have chromosome, from, to, and then gen. So basically that is what we are adding here. All right. Now we also need the header, which we use -h. Okay, so this information we are adding to the VCF file. Maybe it's for comment for some stuffs. All right. So we have that also here. So here, from the instructions in the page, let's get back to the page here. This is how it was specified. Okay, so I'm going to use the same information here. Okay, so -h, and then you have all these. So basically it's the same thing I'm going to do in this command here, this terminal. So -h, my less than symbol, I bring my parentheses, I'll say echo, single quotes, ## info equals ID equals gen, number equals 1, type equals string, description equals gen name. All right, so this is it. You can just copy and paste as well. That's also fine. All right, so this is how it is supposed to be. So annotate, you add your annotation file, your column headers to specify what to carry over, okay, and then your header information. That's what you have here. Now, once you are done, you also need to specify the VCF file that you are going to annotate. So in this case, it's snips.vcf.gz. So space snips.vcf.gz. And then you specify an output file. Okay, so the output file, we still want a gzip file. So I'll say -o, uppercase O, space Z. That means we want it gzipped. Then I'll say -o, lowercase o, and then give the output file name. So in this case, I'll say snips with gens.vcf.gz. So I am making it descriptive here so that whoever sees the VCF file will have an idea of what the content is. Okay, so this is going to be the information. So let's execute this command. Okay, once we execute it, it's going to be done quickly because the information we have here is not much, and so bcftools will work quickly to do it for you. Right. So now if you do an ls, you should see another VCF file, which is snips with gens.vcf.gz. Now we need to confirm that indeed the annotation has been done, the gen names have been added. So what we'll do is to examine the info field. All right. So again, we'll use bcftools. So I'll say bcftools query-f %info/n, and I will specify the file. All right, so like this. So let's execute this command. Good. Now when you execute it, you realize we have this what? Gen equals var. So this is what has been added. Now let's do this. Let's just do this. Let's use the original snips file. This time I'll use head. This is what we had. Now if you do for the newly generated VCF file, this is what you have. So you realize that in addition to what is here, we have the gen name added. So here it says gen equals var. Okay, so why gen? Because when we were calling it, we said what? Gen. This here. So this has been added. Okay. So that's what we have here. So once you have this, then you can do other stuffs. All right. Now this is a VCF file. If you annotate it, you can also export some of the information to a spreadsheet software that you can analyze or view. So for example, let's just say, for example, we want the chromosome, maybe, and then the position, and then also the ref alt, and then maybe the gene name. Okay, there are ways you can use to extract that information. Now I'm going to do just this simple command so that we can extract this information to a text file. All right, let's clear the screen. Let's ls, and then let's do this. Okay, I'm going to export some of the information here to a text file. Okay, so what I'll do is this. I'll say bcftools query-f %, and here I'll say Chrome. Okay, and here I'll say comma, and I'll say post, comma, and I'll say ref, comma, alt. And then I'll add the info. So that's how it's supposed to be. Now, because this is a VCF file, we need to always bring this for each of those fields. And so I'll redo it again. And this time I'll say post, comma, ref, alt, info. And I'll say /n. And I'll say snips with gens. Now, if I do it this way, then I'll redirect to an output file. So I'll just say variants, or you can even say snips.csv. All right, whichever one you want. So snips.csv. Now I'll run this, and if I do an ls, I'm going to see this file here. Now this file, if I do a head, I'm going to see all this here. This file you can open with a spreadsheet software. Let's see how that is done. So what I'll do is to use my file manager, and then I'll come here, I'll open it, and this is it. All right. So this becomes the information. Now for this command, I did not add a header. So that is why you have it this way. But you can also add header using utility like set, okay, to add headers, and you can now check what you have. So we have our chromosome, position, ref, alt, and we have the info field, all right, which has all this information, including the gene names, what you have here. Now, there are other commands you can add to extract just the gene names. It's possible to do that, all right, so take note of that. Now, for regions where BCF tools do not find information, you will see that they are blank. Okay, that's another thing you also have to take note of. So those that are blank, that means that information, there was no information in the bed file that BCF tools could use. All right, so that one, you are responsible for making sure whatever information is needed to be added, you place them in the bed file. All right, so this is how we do it. Okay, so that is a good thing. So today we've learned how to add gene names to variants in a VCF file using BCF tools. All right, so there are other tutorials that you can use to manipulate or handle VCF files using BCF tools, and I have an entire playlist on that. So check the description box and you find the link to watch those videos. Okay, for those who also want to support the channel, I have a Patreon link. You can also buy me a coffee as well. So that'll be all for this tutorial, and I'll see you in the next session.